Hello, and thank you for joining us today for this Boston Cell Standards webinar hosted by Samba Scientific. My name is Jason Amsbaugh, and I'll be the moderator today. Our speaker today is Dr. Stephen Bogan, a board-certified clinical pathologist and adjunct professor of pathology and laboratory medicine at Tufts University Medical School. Dr. Bogan is an inventor on 22 patents, all of which relate to various medical diagnostic technologies. Previously, Dr. Bogan served as medical director of the clinical chemistry laboratory at Tufts Medical Center until 2021, and was also the founder of Cytologics Corporation, which developed the artisan instrument for automated complex histopathology stains and is now sold and supported by Agilent Corporation. In this webinar, Dr. Bogan will discuss new technology for IHC assay calibration and its applications for translational research and companion diagnostics. So without further ado, I'll go ahead and turn it over to Dr. Bogan.
Good morning. Set up. We're all set. Just thank you so much, Jason, for the kind introduction. So this seminar is aimed at sharing really a new understanding of immunohistochemistry testing. The new understanding and really the reason for this seminar is it's driven by a growing gulf, a divide between the level of accuracy and reproducibility that's asked of immunohistochemistry versus what the method is currently capable of delivering. So in this seminar, I'm going to describe how you can avoid the pitfalls of IHC in the context of companion diagnostic assay development in association with drug development, and use these new tools, immunohistochemistry calibrators, to your advantage. The presentation is aimed at industry scientists working across the drug development spectrum because companion diagnostic assay development really starts early and evolves with time. This figure illustrates the parallel development for companion diagnostic tests as a drug candidate progresses through the development process. Establishing a companion diagnostic assay and exploring various analytic sensitivity cut points is appropriately started during preclinical studies. By the end of phase 1 and phase 2, the assay analytic sensitivity cut point that separates stained from unstained cells should be locked in. By the time the clinical validation study started, the assay should be fully specified and locked down, which includes all clinical decision points and any scoring or interpretation. So the next 45 minutes, I'll share with you the following 4 topics: an explanation of floating cut point error, why it's missed using current IHC assay validation protocols and quality assurance, a description of IHC calibrators and the technology, and finally, 3 examples of pitfalls using published data. A floating analytic sensitivity cut point error is unique to immunohistochemistry because in lab medicine, we routinely calibrate assays. So this includes qualitative immunoassays like IHC, but in the clinical laboratory, the blood laboratory, examples include HIV or hepatitis serology assay. In those qualitative assays, we calibrate to establish a reproducible analytic sensitivity cut point or threshold for distinguishing positive from negative test results. It's only for immunohistochemistry that we do not calibrate. Immunohistochemistry test results are evaluated by whether you can see a visible stain. What many don't appreciate is that the ability to see a stain depends on the analytic sensitivity of the IHC assay. Each assay has a cut point or a lower limit of detection. The cut point is the minimum biomarker concentration per cell that is required to see a visible stain. Without calibration, you don't know the cut point. Therefore, a floating cut point is a staining threshold that fluctuates from lab to lab or day to day. Allowing that threshold to land wherever it might, such as illustrated here with the red dashed line, leads to mistakes. In clinical IHC labs, it's mainly false negatives. In this example, about 1/3 of the true positives or drug responders will test negative, false negative, because their cellular concentration is below the cut point to the left of the dashed red line. This illustration depicts a situation where about a third of the true negative population, the non-responders, test positive in this assay. These are false positives. And of course, this would be the ideal for maximum separation of the two populations you're trying to distinguish. My point is that without calibration, you have absolutely no idea where that cut point lies. So let me share this example. This is a breast carcinoma core from a tissue microarray stained for estrogen receptor. Now, I ask you to consider if you would call it positive or negative. In other words, should this patient receive hormone blocking therapy? Hopefully, it will be clear the biopsy shows plenty of ER-positive cells, leading the patient to a recommendation for hormone blocking treatment. Okay, what about this one? Same question. There may be a faint tint of brown in some cells, but hopefully you'll agree that the appearance is largely unstained. This patient would receive a different treatment. Now, the reason for presenting the two carcinoma samples, both stained for estrogen receptor, is that they are, as you may suspect, one and the same sample, stained at two different accredited IHC labs. So here's the two photomicrographs. The one from Lab A is above that of Lab B. The reason for the different test results is that the two labs have different analytic sensitivity cut points, as written along the left, highlighted in yellow. Lab A's lower limit of detection, or LOD, was just over 7,000 molecules per cell equivalent. In other words, the assay only requires a little more than 7,000 molecules of estrogen receptor to produce a visible stain. Lab B's cut point, or LOD, was 10 times higher at almost 75,000. In fact, from a tissue microarray containing 80 different patient samples in this particular national survey, there were a handful of tumors with estrogen receptor concentrations between 7,000 and 70,000 per cell. These are the ones that are positive at Lab A and negative at Lab B. This was the first broad national survey that incorporated IHC calibration in explaining differences amongst IHC laboratories. Those cut points make a huge difference, but only for a subset of cases. This graph shows the percentage of positive cases in the tissue microarray along the y-axis as a function of analytic sensitivity on the x-axis. Each dot is the result from a single participating lab. At the far left on the x-axis, those labs are highly sensitive. They have low cut points or lower limits of detection. It doesn't take many estrogen receptor molecules in those labs to the left to generate a visible stain on the assay. In this graph, we also stratified the low and the high ER-positive cases. So the upper curve in brown, the high positive cases, reveals that regardless of the cut point, roughly 35 to 45% of the cases are positive. So these cases express estrogen receptor concentrations that are above pretty much all the cut points of all the laboratories, so that the exact one really doesn't matter. For the low positive ER cases in this tissue microarray, the cut point has a huge impact on whether they are read as positive or negative. The positivity rates, as you can see, vary from almost 0 to 35%. So the conclusion is that the cut point, or LOD, lower limit of detection, directly affects test results, but for a subset of samples. Here's another example, this one not yet published. The study design was similar. NTRK, an NTRK immunohistochemistry testing, is important for determining eligibility for treatment with a TRK inhibitor, such as Vitrakvi. As the graph shows, clinical sensitivity is directly related to the NTRK cut point. With inadequate sensitivity, the labs to the far right are missing almost half of the patients who would benefit from the TRK inhibitor. So let me transition now to another topic, bullet point number 2 in the sequence. So in this next section of the seminar, I'd like to share with you essentially why the published guidance for IHC assay validation is missing these important aspects. IHC assay validation mainly comprises a side-by-side comparison testing samples in the new assay under consideration to expected results. Usually the expected results are based on a previous IHC assay. The accreditation requirements from the College of American Pathologists, or CAP, describes it here as recommendation number 1. Highlighted in yellow, it explains that new test results are to be correlated with expected results. The remainder of the text explains how you might determine expected results. For example, the expected result may be based on the result in a previous assay in your lab or the result at another lab. So here's an example of a perfect assay validation set expressed in a Punnett square. Test results on the new assay under consideration are compared to sample results on a predicate or previous assay. In this example, there's 100% concordance. All 20 positive test results on the new assay agree with the predicate assay. The same for the negatives. So generally 90% concordance is a passing score. Okay, so now let's compare IHC assay validation guidance to that for assays in general. At the top of this FDA guidance document page is the header Guiding Principles for Bioanalytical Method Development and Validation. There's 10 bullet points which are expanded in the callout. The side-by-side sample comparison I just showed you is bullet number 7 under accuracy. However, I ask you to focus on the first and the third, reference standards and calibration curve. Neither of those exist currently in immunohistochemistry assay validation. Here's another example, this time the laboratory accreditation requirements from the College of American Pathologists. The requirements describe assay calibration requirements on the right for chemistry and immunoassays. The corresponding requirements for immunohistochemical assays are in the left-hand column. And in fact, there are none. Now, this is a huge gap for the immunohistochemistry industry. Assay calibration is a foundational principle in laboratory medicine. In fact, it is the process for standardizing laboratory testing around the globe by linking assays to a primary reference standard. It is required even for qualitative assays like IHC that are reported as positive and negative, because, as I'd mentioned previously, calibrators establish the threshold distinguishing positive from negative test results. Here's another perspective, comparing reference standards between industries. In the broad field of laboratory medicine, there are hundreds of primary reference standards, one for each assay. These reference standards are maintained at organizations such as listed here on the left. In the field of laboratory medicine, immunohistochemistry is the outlier, having not a single primary reference standard. I don't know if you've ever noticed, but there are no conventional units of measure in immunohistochemistry. The table lists several examples of units of measure in laboratory medicine on the left, all of which are linked to a primary reference standard. In IHC, we have some units of measure, but none of them are traceable or linked to the underlying concentration of the analyte. These are relative units based on morphologic descriptions. As a result, mid-range IHC measurements are inherently unreliable. This is an illustration of that point, that mid-range results are unreliable. The data are from published proficiency test results by the College of American Pathologists. Twice a year, the CAP sends laboratories a HER2 proficiency survey. Each survey is comprised of 20 unknown tissue samples to analyze for HER2 status. The labs are required to stain the slides and report their results. The graph summarizes the HER2 proficiency testing data from more than 1,000 IHC laboratories, mostly in the United States, from 2022 and 23, the last year and a half. From 3 surveys, there were 60 tissue samples. Collectively, from the 3 surveys, as you see here in the graph, there are 42 negative samples and 18 strong positive samples. I ask you to take notice, there is not a single mid-range or 2+ sample. Whenever mid-range samples were included, there was so much disagreement amongst the labs that they were not graded. There is actually a rich literature describing the difficulties experienced by IHC laboratories in the absence of calibration. These summary data from one of the leading European proficiency testing programs summarizes the performance of participating labs over more than a decade. The data, in my opinion, are especially impressive because the program incorporates central review of the participating lab slides. Namely, that the same small group of pathologists read all of the participants' slide as a group, minimizing variability in the readout itself. We don't do that here in the United States. And the data show that consistently, year after year, approximately 10% of labs fail or under the poor category, and another 10 to 20% are borderline. These graphs summarize a body of literature. Actually, I reviewed in an article in 2019 on error rates in laboratory medicine, including both IHC and clinical chemistry. In immunohistochemistry, the graph on the left, error rates have remained relatively constant in double-digit levels for 2 decades. Clinical chemistry, once many decades ago, experienced similar problems. As modern quality assurance methods were introduced, the error rates fell to less than 1%. The story suggests that similar improvements in immunohistochemistry can be achieved by following in their footsteps. So summarizing this section, present IHC validation protocols do not incorporate calibration, which is a standard of practice in every other laboratory testing industry. And without it, we're usually accurate for samples at the extremes, but negative and high positive, that is negative and high positive, but samples in the middle are problematic. For the next several minutes, I'll share with you a description of the first immunohistochemistry calibrators that my team developed. Now, the concept of calibration is deeply ingrained in lab medicine, but foreign to immunohistochemistry. So it's worthwhile just to spend a minute on the topic. This ruler can be considered a calibrator. In other words, it demarcates a unit of measure, length, traceable to a primary standard. So it comes down, how do I know that the ruler is accurate? It's not enough to go and check it against another ruler because the other one could also be inaccurate. Rather, a manufacturer of rulers checks the accuracy of their ruler manufacturing equipment against a higher order standard. And in the US, the highest order standard is at the National Institute of Standards and Technology, or NIST, here in the middle of this picture. The question then arises, well, how do I know that the ruler at NIST is accurate? Well, that in turn is checked against a primary standard at BIPM at the top, Bureau of Weights and Measures in France, where the primary standard is kept. For many years, that primary standard was a platinum iridium bar, which was exactly 1 meter long. So then how do I know that the BIPM primary standard is accurate? And we know because we all agree that that's what a meter is going to be. So we need to create a calibrator traceable to a primary reference standard, but suitable for immunohistochemistry testing. So on the left is our illustration of a cell. On the cell membrane right-hand panel is a representation of the thousands of different proteins found on a cell membrane. Now, one of them is our assay target, highlighted with the arrow. So let me focus on that in the next slide. So here's our target. Now, to create a calibrator, we need a cell or something like a cell that's stable for measurement over a prolonged period of time and has a defined biomarker concentration, traceable to a primary reference standard. Moreover, we need to create a calibrator series spanning a range of concentrations to define the analytic response curve. So far, no one has succeeded in creating a calibrator using cells. So my team developed a synthetic cell equivalent with funding from the National Cancer Institute, having defined molecular concentrations as would be needed for a calibrator, and that's illustrated here on the right. Now, what's important here, we first attach a fluorochrome to that macromolecule or a peptide epitope. There's no measurement standard for any of the different cellular proteins, but there is one for fluorochromes. So by attaching the purified, that fluorochrome to purified molecules, we can measure its concentration after it's attached to cell-sized microbeads, which is this kind of gray surface to which you see it being attached to. And this is really a standard system of calibration for flow cytometry. So we end up with microbeads coated with the biomarker, and those biomarkers are fluorescently labeled. These microbeads are the size of cells. They behave like cells during IHC staining. And as I'd mentioned, they're measured against a fluorescence intensity standard at NIST. Actually, this is a description and summary document of NIST, Standard Reference Material 1934. It's the standard to which we trace to. One NIST standard serves for all immunohistochemistry assays. So calibrator beads start out in suspension in a test tube. Moreover, since they're chemically synthesized, we can prepare calibrators at a range of biomarker concentrations. Typically, the concentrations range between around 10,000 to well beyond 1 million molecules per microbead or cell equivalent. The fluorescence serves for concentration measurement, and it doesn't interfere with bright field measurements. As I mentioned, these are widely used, that is, the system is widely used in flow cytometry, so we're able to run these through a flow cytometer, and you can see a series of different concentrations, especially related to fluorescence on the x-axis. This is what stained calibrator microbeads look like when placed on a slide and viewed under the microscope. In this illustration, we co-incubated the microbeads with MCF7 transformed cell line, fixed it, and then stained both for estrogen receptor to illustrate the two side by side. Some microbeads have ER covalently attached to the surface while others don't, so that accounts for why some are unstained. So the microbead calibrators, as you can see here, are perfectly round spheres. There are compelling reasons why synthetic calibrators make sense. Most important, they are the only stable products the size of cells with concentration measurements traceable to a primary standard. We published a series of papers documenting their properties, and I'm pleased to say the first products using this technology, breast cancer controls panel, is now FDA cleared. IHC calibrators are pre-printed on a microscope slide. Here you can see 15 small round spots. Around the periphery of the spots, there are the numbers 1, 5, 6, and 10 to provide an orientation to calibrator levels. The spot nearest level 1 in the upper left has the lowest concentration of calibrator. 10 is the highest. So the middle row is comprised of negative controls, and we provide the exact concentrations for each level on a certificate of analysis. So to read the calibrators, you simply identify the lowest level that's still stained. In panel A above is a schematic representation. Panel B below shows what the calibrators actually look like. In each, level 5 is the lowest that still has sufficient color intensity to consider positive. You then look at a certificate of analysis for that calibrator and correlate it with the actual concentration. With image analysis, you can generate an analytic response curve. The analytic response curve defines the relationship of stain intensity on the y-axis as a function of biomarker concentration on the x-axis. From the analytic response curve, we can then define two important analytic parameters, which are the lower limit of detection and the linear range. The introduction of IHC calibration created the need to offer guidance to clinical IHC labs on the expected analytic cut points for the various assays that are widely used now for patient care. As a result, we founded the Consortium for Analytic Standardization in Immunohistochemistry. It's funded by the National Cancer Institute. The organization is described in this publication from Archives of Pathology and Lab Medicine. The consortium has a website, and that web address is listed at the bottom. Closing out the section about calibrators, this graph is useful in illustrating the difference between a calibrator and a control. Controls and calibrators are made of the same materials. They're both formalin fixed. So to illustrate, here is a typical analytic response curve that's generated with calibrators. A positive control could be a single calibrator with a concentration in the linear range. I've circled it here, as you can see with green. Some prefer two controls derived from samples towards the bottom and towards the top of the linear range. So these would be low and high controls. IHC controls are provided in vials as a liquid suspension. To use them, you pipette 1 microliter onto your sample slide, somewhere not overlapping your tissue sample. And the droplet dries, adheres, it retains the microbeads on the slide surface, and then you process it as you normally would with the deparaffinization, antigen retrieval. The formalin-fixed microbeads behave like cells in an IHC stain. Okay, transition now to really 3 useful examples, I think, of common pitfalls in immunohistochemistry testing. So to start, I'm sharing with you the published PD-L1 analytic response curves of the 4 FDA-cleared IHC assays. Please take notice first how far apart some of them are from one another, the concentrations of PD-L1 being delineated on the x-axis. Especially look at the SP263, which is to the far left, and the SP142 to the far right. So this information is going to be relevant as we go forward. So in this illustration of nuclear IHC staining, I ask you first to start this. In your opinion, would patients A and B be placed into the same or different treatment groups on the basis of these images? In other words, are these stains identical or different? I hope you'll agree both are strongly positive. Whatever the clinical context, these patient stains, at least to me, are completely indistinguishable. Okay, now let me juxtapose. Instead, consider this hypothetical biomarker expression graph with analyte or biomarker concentration on the x-axis. The graph shows the true negative and true positive patient groups. For example, these might be responders and non-responders. And as you can see, I've represented them here to be dramatically different protein level expression levels, as I say, on the x-axis. So now, mostly you'd expect these 2 groups to be readily distinguishable when measuring protein expression by immunohistochemistry. And yet these 2 situations can easily coexist as a pitfall here. Without calibration, groups with strikingly different expression levels can appear identical after immunohistochemistry testing. And there is a very simple explanation. I'm showing the same graph you just saw, but now with a superimposed analytic response curve with stain intensity on the y-axis on the right. So recall that I'm drawing it to simulate the SP263 PD-L1 curve. If the concentration of sample B, let's say, is in the negative group at around 400,000, and sample A, the positive group, is at 1.2 million, they're both on the analytic response curve plateau. The staining will be indistinguishable. Both concentrations are, being on the plateau. This is why the cut points and linear ranges are so important. If you're familiar with immunohistochemistry testing, you may be aware of a commonly used method to try to address this problem. Namely, in many instances, we'll increase the percentage of positive cells before calling the assay positive. So I suggest we explore that as kind of a second pitfall. The slide here illustrates the concept, showing different percentages of positive cells. For ER, estrogen receptor IHC testing, the cut point for treatment is usually in the 5 to 10% range. For Ki67, the recent FDA-cleared assay, it's 20%. And for PD-L1, depending on the assay, it's 50%. Increasing or decreasing the required percentage of positive cells has the effect of adjusting the cut point. The reason it works is because, as you're aware, tumor cells in a single biopsy are heterogeneous. From one cell to the next in a biopsy, they don't all express the exact same biomarker concentrations. From one cell to another, there's a range of concentrations. So by increasing the required percentage of cells that have to stain before you call that positive, we're requiring the cells with higher concentrations to stain. And so this has the effect of nudging the cut point higher. However, any increases to that cut point by this method is limited by the dynamic range of the assay. So let me just explain the dynamic range, going back to this PDL1, since I showed it to you already, the PDL1 dynamic range for SP263 is the analyte concentration that spans from 0 to 100% stain intensity. Here, let me show you on the next slide. I think it illustrates it better. So here's that analytic response curve and the dynamic range. Again, the assay dynamic range is the analyte concentration that spans the 0 to 100% stain intensity range. And that range, again, it's a biomarker concentration range. So, for example, tumor cells that have analyte concentrations below the dynamic range, in this box, cells unstained, and they're unstained because their concentrations are too low for this particular IHC assay. Now, cells with biomarker concentrations above the maximum of the dynamic range, well, they're all stained. They have cell concentrations that are readily detected, stained to the maximum extent. The 0 to 100% percentage of cells staining, that adjustment, only adjusts for concentrations within the dynamic range. If the range is narrow, you're not moving it very much. So let me show you how that might apply to a hypothetical precision medicine context. So in this example, with 2 patient groups, the concentration of an immunohistochemical biomarker from patient biopsies is demarcated along the x-axis. It shows that responders are a distinct but overlapping group from the non-responders based on biomarker expression. If the IHC assay has the indicated cut point, then all the responders are staining because their tumor cells all have concentrations well above that dashed red vertical line. In addition, the majority of the non-responders also stain. They also have cellular concentrations above the cut point. So this hurts clinical specificity. The immunohistochemical assay probably or may not be viewed as especially helpful, and the data may appear, though there's little difference in biomarker expression between responders and non-responders, even when there is. Now, to solve this problem, you might reasonably decide to try a higher cut point. Where you place the cut point may depend on a variety of factors, such as the treatment risk profile. But let me show you what can happen if you try to raise the cut point by increasing the required percentage of stained cells. So superimposing that analytic response curve onto this hypothetical graph, as you can see, the cut point, as you go from 0 to 100% percentage cells stained, it does not shift, or it may not shift as much as you might want. And therefore, you may conclude that the biomarker is still not useful in discriminating responders and non-responders. And the treatment development program may fail when it need not have. In this situation, the answer would have been really to change the assay's design itself. The last pitfall I'm going to discuss is a little later in the development stage in conducting multi-site clinical trials. In conducting such a trial, an important question is how to standardize testing across all the sites. And now remember, hopefully you've already identified the correct assay cut point. You now need to ensure that each of those clinical trial sites their IHC assays all have that same cut point. To grasp just how challenging that is, let me share with you a real example published last year of 5 identical instruments in the same IHC laboratory in a major academic medical center complex. Over time in this lab, concerns evolved about staining inconsistency, stains that possibly looked weaker than expected sometimes. It was intermittent, and on some stains and not all stains. The lab's medical leadership tried a few interventions, but it was still unclear if this ill-defined intermittent variability was solved. So to better investigate, they ran our PDL1 calibrators on each of the 5 instruments that they had. They're identical, the same model brought into the lab at the exact same time. The instrument designations are listed at the top. These designations are derived from the last 3 digits of the instrument serial numbers. The graph here shows the analytic response curves on a semi-log graph. Stain intensity of each of the 5 instruments with the serial number designations in the legend are these analytics, they're graphed as a function, stain intensity is graphed as a function of PD-L1 concentration. The calibrators repeatedly demonstrated that instrument 108 with the orange square data points was an outlier. The manufacturer's service personnel could find nothing wrong with the instrument, but the data were reproducible across multiple measurements on separate days. The instrument was then replaced and the problem was solved. The difference between 108 and the others was actually visually apparent on the calibrators. Stained PD-L1 coated microbeads stained on 108 are on the left, and another good instrument, the 109, on the right. The same publication actually also tested variability from one slide staining position to another, all on the same instrument. They did this by applying a control to each slide as unslide controls. The red arrow indicates a problem at position 3-10. It was an intermittent glitch. The problem did not repeat on the subsequent staining run. So in conclusion for this presentation, there are potentially serious pitfalls associated with current practice of not calibrating immunohistochemistry testing. It is only, in my opinion, a matter of time until the regulations catch up. The most important benefit of including calibration, in my view, is in defining an optimal analytic sensitivity cut point, as stated in the upper left. Without calibration, it's like flying blind in a fog. Without calibration, clear negative samples, strong positive samples, usually those are accurate. It's the samples with mid-range biomarker concentrations that cannot be measured. So thank you for attending, and I look forward to, if I can, to answer your questions.
Thank you, Dr. Bogan. All right. If you have questions, please go ahead and put them in the Q&A box. Once again, Q&A would be the questions essentially that we would have. So we do have a couple right off the bat. The first question here, why did immunohistochemistry testing develop over the years without calibration, especially if it's a standard in other types of laboratory testing?
So thank you. The answer really, calibration evolved in clinical chemistry as those assays started, and it was much simpler. For example, to get a sodium calibrator, we just dissolve sodium into a buffer. For immunohistochemistry, there were no, technically it just wasn't possible. There were no measurement standards to define cellular concentration of analytes. And so there was an attempt about 20 years ago. There was a conference actually, and they published the results of that conference at NIST, that there was an aim to develop the first HER2 standard, because about 20 years ago, that was when the first problems in HER2 consistency had developed. There were clearly apparent, but it was a much more difficult challenge, and that effort at NIST kind of was eventually discontinued. So the reason is just, it has been a very challenging question, a difficult challenge. And I can tell you also from our standpoint, it took quite a number of years in research before we developed this.
Thank you. The next question is, how do your calibrators compare to cell line controls?
Well, there are, so you can buy cell line controls. The difference, I think the major difference is that with calibrators, you have defined concentrations traceable to a primary standard, which is the way, that's how everything is, what that defines essentially a calibrator. The cell line controls are typically sold as high or low with ill-defined or undefined cellular concentration. So you really can't make an analytic response curve from those, defining linear range or lower limit of detection.
Thank you. The next question is there clinical relevance to stratifying patients with quantitative protein readouts rather than just IHC positive versus negative? For example, like a quantitative or IHC low level versus IHC high.
So I'm making sure I understand the question. Is there clinical relevance to defining, what was after defining IHC high and low?
Yeah. So for example, like in the assay, you were discussing essentially in one of those slides, the theoretical responders versus sort of non-responders, I guess, and understanding what that threshold of protein expression is between those two different groups.
Okay, great question. And the answer is most definitely there is clinical relevance, and actually it's already published. So the estrogen receptor study was published in the journal Clinical Chemistry, and that demonstrated that was the first demonstration of essentially the clinical relevance of whether it's positive or negative and how the analytic cut point affected those results. The NTRK data were also, I mean, that was very big news, particularly because the labs that were using a ready, I should say the off-the-shelf assay, the one that's ready to use, were dramatically insensitive, missing about almost half the cases of patients who were eligible for Vitrakvi treatment. And again, so the labs that got it right were using laboratory developed tests. So, I mean, those are just, the PD-L1 story is also another example. In that case, the assays were already FDA cleared, but there had been an extraordinary amount of attention focused on trying to understand how these 4 different FDA assays relate to each other, and particularly because each one is linked to a different immune checkpoint inhibitor. And it was, there are only a handful of labs, most labs will not stock all 4 assays, some of them requiring different instruments. And it clearly demonstrated that these assays are not readily, at least not all readily interchangeable. I hope that answered the question.
Yes, thank you. The next question is, can you create calibrators and controls for novel targets?
Yes, we have actually a process which we follow, and it really depends on what the target is. Sometimes we'll use a recombinant protein like the extracellular domain of PD-L1. It's preferred, it's easier if we can use many current monoclonal antibodies used clinically are to do peptides. They were actually raised to peptide carrier conjugates. And those actually are, they're easier because we can engineer them with exact concentrations of fluorochrome. So we essentially go through a process of identifying what the analyte, what form of analyte we're going to use, and then we have a standard manufacturing process that we go through afterwards downstream in assembling the calibrator.
Thank you. Beyond, I guess, sort of products, how are you working to educate pathologists, inform policy, in terms of just trying to get adoption, I guess, of these calibrators and controls in current clinical use?
Well, so that's an important, and one answer, of course, is webinars like these. The consortium that I previously mentioned, education, informing the pathology community, is a major task, a challenge, a mission for the consortium. And that will really start to kick in once we've published the very first studies. There are several right now, there's 4 studies underway right now. There'll be several others underway launching this summer. But once those come to fruition, once those get published, then it'll be the mission of CASI, the consortium, to publish and let everyone know what the guidance will be.
Gotcha, thank you. Have you seen interest already with regards to CDX or clinical assay developers from diagnostic companies in terms of using this in their process?
Yes, it kept us busy. So for example, in 2023, we've manufactured, I think it's somewhere around a dozen different calibrators. Some of them for the consortium, some for CDX manufacturers or pharma or CDX manufacturer.
There's a question here around assay variability. So the question is, for drug development, when should we transfer manual IHC staining into automated IHC staining? So for example, sometimes we might get very different results due to amplification systems using automated IHC systems.
Good question. You know, there's no one answer to it, but let me answer it in a different way. If you're using manual IHC or whatever the limitation, if it's manual or if it's just something else is not quite in the final version, the detection system, for example, the key part of it is if you find in preclinical that it's effective, that you're seeing distinction between the two groups that you're trying to distinguish, you need, regardless of how you change the assay, such as automating it or putting a new detection system in, you need to be aware of the cut point. You need to be aware that if your analytic sensitivity changes as you transition to the next stage of development, you will not get the same answers. And so I would urge you not to so much focus on, yes, you are changing the method, but it's in that transition, how have you changed analytic sensitivity? Because if you do, you are at risk of getting completely different results.
Thank you. And then this is maybe just a recap question. We're just about out of questions. So if people have additional questions, please go ahead and submit those. So the question here essentially is, what do you think is the greatest innovation in this final product? So really in terms of just the user needs, I guess, that you had sort of identified and maybe spoken to in terms of developing this product.
I'm not, what is the greatest innovation in developing this product? Did I get it right?
Let's maybe take it a different way and say, what do you think is the greatest impact, essentially, of having this tool in your pathologist toolkit moving forward?
Oh, got it. Okay. You know, it's strange. Right now, there is, I'm coming at, maybe I'll preface it with this little explanation. For my clinical responsibilities, which mostly are, I've handed off to others, but I've been a clinical pathologist, medical director of a clinical chemistry lab. And all my research has been, though, relating to pretty much anatomic pathology methods. Laboratory professionals, often when we do research, it's in developing better methods, assays. And so even though I'm coming from a clin path background, my research and development efforts have been focused on anatomic path. And so the impact really is going to be, I believe, making the assay validation and quality assurance systems of IHC, of an anatomic pathology discipline, to be resembling clinical pathology. As I showed you the checklist questions, for example, of immunoassay and clin-chem compared to IHC. Right now, there are these two very distinct intellectual silos. And I'm hoping the impact will be to blend those two.
Excellent. Thank you. All right. It doesn't appear that there are any more questions. So with that, I'll go ahead and wrap up today. Thank you again for attending our webinar today. If you have any additional questions about these technologies or services or products, please visit bostonsellstandards.com. So thanks again for tuning in today. Thank you, Dr. Bogan, for all your for your presentation, as well as for everybody else for attending. And with that, have a great day.